Right. Living environment. Next year it's not going to be called living environment. It's going to be called life science, biology. Yeah, they can change some things. So, next year. But that'll be next year. You guys don't have to worry about that. You'll be in Earth and Space Science next year, most of you. So you'll enjoy that, I'm sure, with either Mr. Sexton or Mr. Bucher or whoever might be teaching that class next year while you're in the 10th grade. But let's do today's bell ring. All right. Hey now.
Mr. Kovacs class. It's Mr. Kovacs class.
You might learn a ton.
It's Mr. Kovacs class.
He's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class.
Today's Bellringer, I wanted to go back and check some of those Regents questions from the midterm that were really difficult and see if you finally got them now, okay? That's what we're going to do during this Bellringer time. So let's get to it, all right? The diagram below represents a protein molecule present in some living things. This type of molecule is composed of a sequence of A, amino acids arranged in a specific order, B, simple sugars alternating with starches arranged in a folded pattern, C, large inorganic subunits that form chains that interlock with each other, or D, 4 bases that make up the folded structure. What's the best answer choice? That's right, it's A, right? Because protein molecules are made out of amino acids. A. Good. Diagram below represents a cell and some molecules in its environment. Which molecule would require the use of energy in order to be brought into the cell? Is it A, molecule number 1, B, molecule number 2, C, molecule number 3, or D, molecule number 4? That's correct. Molecule number 4 is the right answer. Right? It's the only one that would be going to get into the cell, would be going against the concentration gradient. Therefore, would need ATP energy in order to function. In the experimental setup below, which substance would be used to prove that the gas produced by the yeast in a vacuum bottle could change the pH of the liquid in the flask? Is it A, a salt solution, B, a chemical messenger, C, an enzyme, or D, an indicator? Correct, it's D, an indicator. Indicators change color, right, when conditions change. And we used an indicator. Actually, the indicator you'd use here would be bromothymol blue, which is an indicator we used when we looked at photosynthesis, and we noticed that it went from yellow to blue, which showed that the plants were using up carbon dioxide. And really what was happening is because the carbon dioxide was being absorbed, the acidity was changing, and that's what the chemical was responding to. The diagram below represents 3 sections of a cell membrane showing 3 different methods involved in transport of various molecules across the membrane. Using information from the diagram, state one reason why the movement of molecules in method C requires active transport. That's right, because it's going against the concentration. It's going from fewer circles to more circles. That requires energy. All of these other ones go from more to less, right? They're going to move. That's my old example of closet full of basketballs. You open the door, they're going to fall right from where there are more basketballs to where there are fewer basketballs. To shove more basketballs into the closet, you need to use energy. The old thistle tube, a concentrated starch solution was placed in a thistle tube with a semi-permeable membrane covering the wide opening. It was then placed in a beaker of water. The height of the solution in the tube was measured every 5 minutes for 25 minutes. The setup and the data collected are shown below. There's the setup. There's the data collection. Right, it obviously increased. Explain why the height of the solution in the thistle tube in this given experiment increased during the 25-minute period. Good, because water moved from the beaker into the thistle tube by osmosis. It was trying to balance out the concentration of starch on both sides. All right, there are lots of resources out on the internet, which are like Regents Review things, and it's never too early to start those. Because we'll do those in class as well, like we've been doing all year, but it's obvious that most of you need a little more work before you're ready to tackle the Regents exam in June. So let's start the second half of the year, and let's make it a point to get a little more studying done, a little more preparation done, and that's as much you as it is me. All right? All right, let's have a good day.